It's recording. So I'll give the intro in Portuguese, Elodie, so the students can get it. And in the end, if they have questions, if they don't want to speak in English and they feel comfortable, I can translate as well.
Sure.
Boa tarde, pessoal. Então, hoje a gente tem a professora Elodie Briefer. Ela é professora na Universidade de Copenhague, na Dinamarca, e ela tem uma parceria com o nosso grupo de pesquisa aqui, o Ecomar, né, pela UFRJ, com um projeto com as baleias brudas, mas ela é bióloga formada pela Universidade de Paris Sul, e ela fez um mestrado e o doutorado em Biologia Comportamental na Universidade de Paris Norte, depois fez um pós-doutorado na Universidade de Queen Mary, na Inglaterra, outro pós-doutorado na Universidade na ETH, na Suíça, né, que é uma universidade lá, onde ela também ficou um tempo como pesquisadora sênior, e hoje em dia ela está na Universidade de Copenhague. Né, os interesses de pesquisa dela são variados, ela trabalha muito com a questão da expressão das emoções, que ela vai falar um pouquinho hoje, mas também com a questão de bem-estar animal, cognição e bioacústica, e trabalhando muito aí com os animais domésticos também, né. Então a palestra dela é bem interessante, eu conheço ela já tem um tempinho, desde um congresso de bioacústica aqui no Brasil. Ela foi boa amiga da minha orientadora de doutorado também. Então, eu espero que vocês aproveitem a oportunidade e, por favor, perguntem, né? Participem. Se vocês não quiserem perguntar em inglês, a gente pode traduzir também para vocês. E é isso. Thank you, Elodie. You can start.
Thanks for the introduction. I didn't get everything, but a bit of it. At least places where I've been indeed.
Yeah.
Thanks for the invitation and presentation. So just please make sure you interrupt me if something goes wrong, because I cannot see the screen at the same time as what I'm sharing. Although I can actually make that a bit smaller, so I see both. Great. So I can see if people follow. And if you're still here and I'm not talking to just myself. Good. So today I will give you a lecture on my main research interest, which is looking at how animals express emotions. I've been mostly focusing on ungulates, so it's going to be a lot of farm animals and also wild animals, so wild ungulates. And then looking at basically comparing species to see how different species express emotions, if they do express emotions in the same way or not. If it's the same way as we do express emotions or not, and if we can kind of understand each other eventually. So we're going to look at first some background, because I'm not sure exactly if you had some lectures on emotions, but it's kind of a complicated things to study in animals. So I'm going back to definitions first. Why should we study animal emotions? Why is it important? And then a bit of the methods, because to understand what I will tell you later on vocal expression of emotions, you first have to kind of understand how do we study animal emotions. And then that's probably good for you also if you're interested in the topic, then you can go back to these slides and look at the methods of how do we study animal emotions. It's kind of a condensed version, because I have only about an hour today. So we'll see how far we go. But the first part would be on why do we study animal emotions? What are the main methods, a bit briefly. And then what do we know about vocal expression of emotions? So what are the mechanisms through which animals express emotions, and what is the evidence? And then I'll end up with a case study. So this comparative work I've done, where we looked at both vocal expression and perception. I think today we'll have time to look only at expression, but I will share my slides where you can see also the perception side. So when we tested the animals to see if they differentiate, for example, expression of emotions from their own species, from other species, from human voice, etc. At some point, I have a small quiz, which would be pretty funny for the students. So you'll have to log in this website. I will give you the link again later on, maybe put it in the chat. So you can log in there, and then I will play some sounds, and then you'll have to guess which ones are positive or negative. So just be prepared. Good, so let's start with why and how do we study animal emotions. And then first, to give you a bit of background and for you to understand why we still know so little. I mean, we know a lot and a lot more than before, but we still know quite little about them animal emotions. And why is that? Is because animal emotions have been regarded as something that is taboo, that we scientists are not allowed to look at for more than 100 years. Because at that time, so after Darwin, so in Darwin's book, he was looking at animal emotions, comparing that to human emotions. But then after that, this became taboo because people thought that science should rely only on observable phenomena and not hypothetical entities like emotions. So basically only on things that we can clearly see and assess. And also animals were kind of considered as machines that just turn on and off. So emotions were not important to, or animals were not thought to have emotions anyways. But then there has been a huge increase since the 1990s, as you can see on that figure with the number of papers published over the years. And that's only 2010. I think by now, more than 10 years later, we are way above that number. There is a huge amount of research done on animal emotions. So very large increase. Nowadays, people even study emotion-like phenomena in insects. And this big push has been driven by pharmaceutical development. So people wanting to develop medicine to treat, for example, human mental disorders, or people looking at neuroscience and trying to treat human mental disorders by looking at brain regions responsible for emotions. So basically, people were using animal models to develop these methods and to study where emotions are encoded in the brain, basically. But then they were still claiming that animals don't have emotions, which is kind of a big mismatch. So that led to a big push in, okay, how do we actually assess animal emotions? And another big push has been the field of animal welfare, which is nowadays clearly defined as being based both on how physically healthy the animals are, so make sure they are not sick, but also how mentally healthy they are, so to make sure that mentally they are doing well and they're expressing positive emotions. So with all these fields, especially the field of neuroscience, now we know much more, and that's what I will explain you in the rest of the lecture. This fall, there should even be the first textbook on animal emotions, which will hopefully be published by, I guess, September this year, which is going to be textbook for students explaining how animal emotions can be studied. But especially the field of neuroscience has explained a lot about animal emotions. For example, this researcher called Panksepp has been mapping what he called the basic emotional systems, which are defined as unconditioned emotional response systems of the brain. And he's been mapping mostly in rodents where the key brain areas for these systems are and what are the key neuromodulators. So for the system called seeking, rage, fear, lust, care, panic, and play. And basically, this research has shown that these basic emotions are encoded in ancient caudal medial subcortical regions, which are shared between all vertebrates. So basically, this means that all vertebrates have the capacity for experiencing these basic emotions at least. Of course, not going into more cognitive emotions such as empathy, etc. So at least these basic emotions, it's a fact, they're shared by all vertebrates. Now, in terms of importance, again, for studying animal emotions, it's important for many things, as you've seen. So for developing, for example, animal models of human mental disorders, develop medicines, but also for the field of animal welfare. As I mentioned before, it's now clearly well established that it's based both on how physically healthy the animals are, but also how mentally healthy they are. And that's also the public concern about animal welfare is strongly linked to the fact that animals experience mental states. And so past research, let's say in the 1980s, 1990s, has been focused a lot on negative emotions. So claiming that we should prevent animals from experiencing negative emotions. So we know a lot about negative emotions and the intensity of these negative emotions, for example, stress. But we still know quite little about what we call the valence, and I will go into more details later on about that, but basically how positive versus negative emotions, how we differentiate between an animal that is in a negative state or a positive state. For example, in that picture, whether this animal is depressed or is relaxed. And that's actually quite difficult to tell, even nowadays. So the main challenge is basically differentiating between negative and positive emotions to promote positive welfare and make sure animals are experiencing positive states, such as happiness, for example, in humans. Good. So, how do we define actually emotions? What are they? So first, they often define as a state, so internal state that is has a valence, so is basically either positive or negative, has some kind of positive or negative connotations. And we can differentiate between emotions and moods. So emotions are the short-term states. So for example, experiencing short fear or short happiness. And these emotions are linked to specific events. So if you imagine yourself walking in the woods, and then you will see a leopard, for example. So the specific event of seeing a predator coming towards you will drive a short-term emotion, which is very likely fear. And then this can accumulate over time to become moods. So an easy way to see that is, for example, you wake up in the morning, you don't have any more coffee, you fall off your bed, you miss your boss. So you experience all these short-term negative emotions, which will make you very moody and probably pessimistic about what will happen during that day. So you'll develop this long-term negative mood, or if you have lots of positive things happening to you, more long-term positive mood. So moods, on the other hand, they are more diffuse, so they're not linked to one specific event, but they arise through this accumulation of short-term emotions. And that makes a lot of sense for animals. For example, a mouse living in an environment with lots of predators will experience lots of short-term negative emotions, and that will develop into long-term mood, which will drive the mouse to expect more negative things to happen. So if the mouse, for example, sees a shadow or something a bit ambiguous, it will logically think, oh, that must be a cat, because that's what it can expect in its environment. But on the other hand, if it lives in an environment with no predator, but lots of food, it will experience lots of short-term positive emotions and then become kind of a more optimistic, so a more optimistic mouse, and expect ambiguous stimuli to be more of food than a predator. So basically, emotions facilitate response to stimuli, should animals approach them or avoid stimuli. And then moods inform about expectation. Should the mouse expect more predators around or more food? So they're actually very important in the life of animals, and they make, so when you understand that, it makes complete sense that, I mean, animals could not function without emotions in their environment. And affective states are also a multi-component response. They're actually made of 4 components. So if we think back again about you walking in the woods and seeing a leopard, you will basically first have change in your heart rate. You'll start breathing faster, your temperature will change, and then your brain activity as well. And then you will likely first freeze and maybe scream and then run away. So you'll have behavioral changes. You will also pay attention more to danger around you. You'll probably memorize this event very, very strongly because it's very salient. And then you will interpret all these changes in your body based on your experience, likely as fear. So you'll consciously process all this information and say that you are now subjectively, like consciously experiencing fear. So in animals, we cannot access these subjective components, but we can measure all the rest. So we can basically measure how they behave, how they process and memorize, and what they pay attention to. And then there are also changes in heart rate, etc. So this is kind of what we can do, but now I'll go a bit more details about how do we study animal emotions, because we cannot directly ask them like animals, how do you feel? And I just want to mention that's basically the same challenge as researchers working on previable babies have. So it's not only people working in animals that have this challenge, it's also people working in human babies. So there has been a lot of framework developed to do that, especially in the past 10 years. There has been a huge explosion of research in the field, and then people trying to map how we can actually do that. And one very good paper cited here has been trying to map what people do in humans, so what psychologists do, and trying to see in a mirror how we can do that in animals. And I will go through that step by step. So first, I mean, we have specific context generating emotions. And if we know the biology of the species, we can predict if this context should lead to positive or negative emotions. We can use this concept that I will describe later on. And then we can also use the indicators that I mentioned just before. So the behavior, the neurophysiology, etc. So first, as mentioned, we can use the contexts. And this is actually pretty straightforward because we can use what is called intuitive inference, knowing what in a species should enhance fitness. And this is usually known if we focus on wild animals. So if we know an animal well, we will know what should enhance its fitness and what usually would threaten its fitness. For example, social behavior versus predators. And we can also test to see what they usually approach or avoid, and then conclude about whether this should then lead to a positive emotion or negative emotion. So positive emotions are emotions that trigger approach towards stimuli that enhance fitness. Negative emotions are those that will trigger avoidance behavior from stimuli that threaten the animal, the fitness of the life of the animal. And then I will go just briefly, because that's quite complicated to explain, but about the concept that we can use. So when we study emotions, we usually will do that within a kind of umbrella framework. And there are two main ones. So some people consider emotions as discrete entities. So they will just consider that there is happiness and sadness and fear and depression, and that all of these are kind of separate, distinct entities. And the other people, like what I would use in my research, and there are lots of people working in animal emotions use, which is called the dimensional approach. So instead of talking about sadness and happiness and fear and considering these as separate entities, we look at the emotions more as a continuum. So basically, we'll consider that emotions go from across two main dimensions, which is, and I will mention that again later, the valence goes from positive to negative, and the arousal, which is how intense the emotion is. So the discrete emotions are, for example, the Plotnik wheels of discrete emotions. So we can consider emotions as joy and trust and fear and look at all these separate emotions. Or what I mentioned before, these basic emotional systems, such as seeking, rage, and fear, which each have their brain region and the neuromodulators. This theory has some advantages because it's actually quite easy to map these labels to specific state of animal welfare. For example, if we see an animal playing, doing lots of care and loss and seeking, then we can assume that it's in a good state of welfare and vice versa. But there is also a big disadvantage, which is that there are lots of differences between languages and cultures in these labels. And also there is more risk of anthropomorphism here, where we would think that the animal is experiencing fear where it's not. And there is also emotions that humans might have, but animals might not, but also emotions that animals might have experienced in their life, which we've never experienced and could not describe. So one approach that is maybe more straightforward, which is what I use, is to divide them simply. So as whether they are negative, so when they are part of the avoidance system, or whether they are positive when they are part of the more approach system. As I mentioned before, so all the negative emotions will usually trigger avoidance, the positive emotions approach. So that's one of the dimensions, which is the valence. And then the second dimension is the arousal. So that's basically how intense is the emotion, because it can be low within the negative emotion. So you can have an animal that is the equivalent of sad or depressed, or very activated in the negative, which would mean fearful, anxious, stressed. And you can have the same in the positive emotion. So you can have an animal that is has a low arousal, positive, relaxed, calm, or high arousal, positive, for example, very excited or very happy. So that's the two emotions that I will talk about a lot in my talk. So the valence and the arousal. So valence is negative, positive. Arousal is kind of the activation of the animal. So the advantage of this method is that it avoids a bit the risk of anthropomorphism. And disadvantage is that some emotions might not be able, we might not be able to differentiate them. For example, fear and pain might actually be exactly in the same spot in these two dimensions. So we might not be able to know if an animal is experiencing fear versus pain. And I don't have enough time today, and also it's more straightforward on Zoom, for example, but I would usually try to ask people, and you can maybe do that on your own at home later on, where you would place these terms to see if you understand this framework. So I'm just jumping to what usually happens. So I ask people, where would you place satisfied? Where would you place how you feel when you're relieved, or when you're bored, or when you're angry? And so here students usually map, do a lot of things. There's huge individual variation. People would usually say that satisfied is, of course, positive, but maybe quite average arousal, while relieved might be more higher or lower, depending on people. Bored is usually something negative, maybe a bit lower arousal, and angry would usually be quite high. High activation, very negative. But there's lots of variation between people because these terms are quite subjective. And yeah, don't hesitate to stop me if you have questions, so you can interrupt me. So that's for the frameworks. Now if we move to the indicators. So as I mentioned, we cannot ask animals about the subjective components, or how they consciously perceive emotions, although they likely do, but we cannot access that, that component. However, we can access the other components of emotions. We can, especially nowadays, clearly measure how the brain activity is changing, how the heart rate and respiration rate changes. We can see how they appraise stimuli, what they pay attention to, what they memorize, how they judge ambiguous stimuli. And of course, as you likely know, we can assess your behavior, mainly their ear positions and head position and movements, etc. So we can use all of these indicators. And I just want to spend also a few more slides on how do we know, okay, which one to use. So should we use behavior or cognition? Like which one actually makes, tell us about the emotions of the animals, and which emotion? So here again, what I usually do when I have more time is that I ask students to think for a few minutes. For example, if they encounter this animal, how would they know whether this, what looks like this smile indicates positive affect in this crested macaques as it would in humans? And then so you can think for yourself a bit. So how would you do that? Which experiment would you do? Which indicators would you use? And so for time management today, I will just show you the methods. But that's what is called construct validity. And just to explain a bit about the background of these methods, so in this textbook on animal emotions that will hopefully come out this year, that I mentioned earlier, Georgia Mason, Mike Mandel, and Liz Paul have a book chapter, which I had the chance to see in advance, where they basically go through these methods. So that's basically my interpretation of their work, which hopefully you will be able to see for yourself soon in this book chapter. But basically, they looked at what people do and then kind of gathered 5 main methods through which that you can use. Ideally, the best would be to use several of these methods. So the first one, so basically, how would you test if it's a smile? And then which indicator would you use for that? So the first one is the most straightforward, of course, the easy one. You go to the literature, you go online, and then you look for papers. And then if you're lucky, if your species has been well studied, you will find the right paper explaining you if this smile is indeed has been described as a positive link to positive emotions, or if you can maybe use some other indicator that will tell you when the animal is happy or in a positive or negative emotion. So you might look at this main review, for example, or you might find, if you work on pigs, clear indicators. You could find the same in sheep, or you could find, for example, the same in cattle. Farm animals have been studied a lot, so if you work on farm animals, that might be easier. Make sure also that, so be a bit critical when you read these, because some people would use a circular argument, which could be, I will use this smile to identify positive situations, and then I see that this smile increases in positive situation. Oh, so it's a good indicator of positive emotion, which is completely circular, so be careful. But basically, that would mean for the question we had in mind with the smile, there is clear evidence suggesting that smile is associated with positive emotions in published papers, in the literature, in this species. So I can use smile as an indicator. It's a good indicator. And then another one is to use basically what is found in humans, which of course, as you can imagine, has lots of shortcomings. So it basically do like Darwin did. These expressions shows, I think it was fear or anger. This one is a smile, so it actually works. It's a good indicator in humans, so it's the same in animals. And this one, which I think was anger, is also a good indicator of the animal being angry. So here you would say, okay, humans in a state of positive affect show increased amounts of smiling, while when they're in negative affect, they show decreased amounts. So I can just assume that smile in my species, which was crested black macaques, is also associated with positive emotions. As you can imagine, it has a lot of assumptions. First one being that in humans, we do have precise and truthful self-report, which is not always the case. And then the second one is that things are similar between humans and animals. So an animal showing a smile also is actually in a positive state, which is often not the case. We know now that a lot of primates, actually, these smile-like expressions indicate fear, not in the crested macaques, actually, but in many other species. The third method would be to look at if animals avoid or approach stimuli, like I mentioned before. So you could basically look at what the context in which the animal is. Is it a situation that it prefers, that it approaches? Is it a situation that in the wild would promote the fitness of the animal or not? And then you will see if in this context that promote fitness, the animal is smiling more or not. So basically, individuals in preferred situation that promote fitness will show an increased amount of smiles, while in aversive situation or fitness that situations that threaten the fitness, there will be decreased amount of smile. Then you will know that smile is a good indicator of positive emotions. Here, the assumptions is that affective states have evolved to promote fitness, which is actually an assumption, although very logical. And that positive emotions promote approach, so approach, seeking, while negative emotions provide avoidance, which is very often the case, but not always. The fourth method is actually quite straightforward. That would be basically using, if it exists, affect modulating drugs. So let's imagine that you have a drug that will promote a positive state in the animal, and you know that it's the case. then you can inject this drug and look at whether the animal is smiling more or not. So individuals given drug that will promote positive affect will show increase amount of smile, that will be your prediction, while those drugs that decrease positive affect or trigger maybe a negative affect will show decrease amount. Of course, we don't always have these drugs available for the species we work on, and the assumption is that drugs have a similar effect across species. Very often we know that that's the case in humans, but we assume that it's the case, only assume that it's the case in animals. And then finally, the fifth method would be to look at which measures co-vary with other indicators that we know are good indicators. For example, individual displaying other signs of positive affect, which could be having the ears in a certain position that we know indicate positive affect. And then we look at when they do that, do they also smile or not? There are also lots of assumptions here that we have. These will validate in the indicators. And that's, of course, that the indicator we're looking at, for example, the ear position that I was mentioning, that they have the same time course. So if you, for example, consider as a well-established indicator cortisol changes, you know that there is a circadian effect. It changes over the day. It might not change, the smile might not change in the same with the same course throughout the day. So I know that it's a lot of information. Please feel free to go back to the slides afterwards. But basically, there are 5 methods you can use to know which indicator to use in your species. If you study a well-known species, you can use indicators described in the literature. If not, or if you're studying a new species, species that hasn't been looked at, and there are a lot, of course, especially white species, you would ideally use several other methods. You would see, okay, what can we see in humans? When does my animal, which stimuli does my animal prefer or avoids? Which stimuli do, in which context are fitness boosting? Which context are fitness harming? And then see in this context what happens to the indicator you're planning to use. You can see if you have access to some affect modulating drugs, and you can experiment on the animal, then you would use these drugs and see what happens to the indicator you want to use. Or you can see if the indicator you're planning to use co-vary with another well-known indicator. And of course, you'll do that in a framework. So basically using either the discrete approach or the 2 dimensions that I mentioned earlier. So now, yeah, I think I really have time for questions afterwards. So I'll just continue for now. But now I'm going to move on basically to a more detailed indicator that we're interested in today, which is expression of emotions. So I mentioned that we can use many indicators. One of them is the behavior of the animals, which could be ear position, movements, locomotion, all these things. But one indicator I haven't mentioned yet in details is basically, as you can see in this picture, clearly the expression of the emotion. So, which can be, for example, facial expressions, but also what I'm mostly interested in, vocal expressions, so vocalizations. So expressions in animals of emotions, they are very important because they will regulate social interactions. They will tell the others, you should back off or you can come closer. So they are aimed at being perceived by others, which is a good thing and a bad thing. First, a good thing because that means that we can likely also see it and measure it. That's something that should be obvious, that we can perceive. But the other is that there is also some intentionality behind, so it might not exactly reflect the internal state of the animal. Some examples of expressions. So you have visual expressions, which lots of colleagues are working on, facial expressions. You can also have maybe chemical signs. So for example, pheromones and vocalizations, which we're gonna now look at in more details. And so when we consider vocal expressions, we can look at which type of sound is produced. So is the animal producing, so is the dog, for example, producing a growl or a bark or something else? But also how it changes, so how this growl, the structure of this growl. So what is this duration? Is it a very long growl or short growl? Is it a high pitch, low pitch growl? And is it very loud or not loud? So that's the two main aspects we can look at in vocalizations. So let's look at a bit about what do we know in terms of mechanisms and evidence for these expressions. So in terms of mechanisms that we can think of, first linked to call type. So how is an animal, a dog, producing a growl versus a bark, for example? And this we can clearly assume that it happens at the level of the brain. So basically we have a stimulus, for example, another dog coming close by, that will change, affect the brain, mostly sensory, emotional, and homeostatic brain areas that will then impact on the brain vocal control pathways. And then through the vocal apparatus, so the lungs and vocal folds and vocal tracts, we will then have the production of a call, so a growl or bark. And likely whether it's going to be a growl or bark that is produced is defined here in the brain. And we know that from research, for example, on pigs, where these researchers have injected, so here we look at the methods that I told you about injecting drugs, so using drug affect modulating drugs. So here in that case, they, for example, injected acetylcholine in the amygdala of pigs. And then they looked at which sound is produced. So when they inject acetylcholine, what happens is that the calls that the animal is produced look very much like screams in the structure and differ a lot from pig grunts, which are usually produced in more relaxed states. So based on that, they know that, okay, when acetylcholine is here in the brain of the pigs, then we get a scream and not a grunt. So that's clearly basically showing that at the level of the brain, when there is fear, pigs will produce a scream. But there's also a huge amount of evidence, so not only for negative calls like I just showed you, but also positive sounds. So one very good type of positive sounds is what we call laughter-like sounds. They've been studied a lot in apes. I can play you this sound. So that would be a gorilla being tickled, or a chimpanzee, bonobo. And basically all these sounds, they look they're very similar to human laughter. So for example, baby laughter. They're very sharp, short sounds that are repeated. And there are also other people that look at play calls, and there is lots of similarities in these sounds. Another extremely well-studied example is rat ultrasonic vocalizations. Maybe you've heard about. So rats, when they're in a negative state, they would usually produce calls at 20 kilohertz. And when they're in positive states, for example, being tickled, these sounds will be at 50 kilohertz. So it's like two different call types, one clearly linked to positive emotions and one clearly linked to negative emotions. In farm animals, with some colleagues, we try to map also what happens. So in positive states, you will usually hear clear types, call types, for example, grunts in pigs, like very short grunts, nickers in horses, etc. I'm not going to play them all. Clucks in chicken. So you have some call types that usually are produced in either negative or positive states, so very specific context. But then at the level of the vocal apparatus, you can also have lots of changes. And why is that with emotion? So why is that? Because of the way we produce sounds in mammals, and also that's very similar in birds. So usually we have air coming from our lungs that makes our vocal folds vibrate. And then this will define how low your voice is, so the pitch of your voice. And then the source sound will be filtered out, so you'll have some modification in that sound that will amplify some frequencies and dampen others. But as you can imagine, if you're, for example, stressed because you have to give a talk in front of everyone, you'll be more tense. So your vocal apparatus will be tensed. You'll be faster. Your salivation will decrease. Maybe the way you speak will change also. And all of these changes linked to your emotions will then affect your voice as well, of course. And we can also predict the way these changes will occur, especially with the dimension arousal. In animals, we can measure the same similar parameters. Of course, they have less capabilities for articulation, but they can open and close their mouth. And we know that how much they open or close their mouth changes these dark lines that I showed here, which are called formants. So, and we also know that they have less intentionality, less control over the voice, and they cannot really fake their emotions. I mean, some of them can, animals that have vocal learning abilities, but many of them, especially farm animals, they cannot really produce a new sound or change the way they sound. So they will basically not be able to hide much of their internal states. So that leads me to my quiz. I hope you'll follow with that. So now kind of that's an introduction to the case study. I'll show you afterwards. But basically, do you think, so when we've looked at different types of calls, for example, growl and bark and laughter-like sounds. But now if we focus on one type of call, for example, contact calls that are produced all the time when animals are being reunited or separated. So when you have one specific type of call, do you think its structure changes depending on the emotion base? So does the duration, the pitch, extra changes? And so for that, I would want you to go to that link. So I'll try to see if I can, oops, sorry, maybe I will type it in the chat and then you can log in and then answer the questions. So where's the chat here? HTTPS. Can you just copy it? So I'll just type it. I hope I didn't make it creative.com. Students. That looks correct. Yes, so you go here and then the password is vocal. So you can go from your phone or computer and then I will basically play some sounds and then you'll have to guess which one is positive or negative. So I'm going to check if people manage to log in. And while you're logging in slowly, I will just show you basically how it works. So I will show you several species. I think we have 6 species in total. And then each time I play 2 sounds. So I will play sound A and then sound B. And then I will ask you which one do you think is positive. And then you'll have to click on A if you think A is positive or B if you think B is positive. So it's always the same question. Which one do you think is positive? Meaning that the other one was produced in a negative context. And it's always the same call type, so it's very hard. For horse, I'm going to play you only whinnies, and then for pigs, only grunts. But one of these grunts is positive, and the other one is negative. So let's see if there are more people here. I see only a few students, but we can slowly start. So it works that way. So I'll play you one sound A and B. And so the question is, can you guess if A is positive
Elodie,
and B is positive? Yes.
The sounds like they were really quick, so I couldn't actually properly hear them.
Okay.
Like
so
before, I could hear them, but like in the other slides, but this one's no. Like I couldn't barely hear A, and B was super fast.
So try again with A. Is it better?
No, it's not coming through. I don't know why. And the other, like on the previous slides, I could hear the sounds, but these one, no. I don't know
what could I do? Not sure. How do I share the whole? Send something else. Windows, screen, pop, and I will see if I share to share audio, share a tab instead, but that's what I don't get. Share screen, Chrome tab. Yeah, but that's only. I'm just gonna try one thing if I share the slide. And then, but how can I? Oh. Let's see if it works better. I know I cannot even hear them myself.
Yeah, I couldn't hear it as well.
Now it works?
No.
No?
No.
Oh, okay, that's a shame. But yeah, looking at the time, I think I'll move on. Yeah, what I can do is that I can share my PowerPoint afterwards with you, Gabriel, to send to the students so they can click on the buttons and try themselves. Yeah, so I'm just going to move on. But basically, so and that's towards. I mean, I have the answers of the large questionnaire we did about that at the end of the slides that you can also look at. But we did a very big questionnaire where we tested 1,000 people and then try to see if they can guess which sound is positive or negative and which one is high arousal or low arousal across all these species. And basically, people can guess above chance level for most species, especially the domestic ones. But it's quite tricky, and they can do that better for arousal than valence, and it's also linked to how empathic people are somehow. So the more empathic someone is to someone else, the more they can guess, the better they can guess. So if we look at, so basically which parameter do we hear? So when we hear these sounds, so if I hoped you could have heard them, but basically you can hear differences between, for example, whinnies or the grunts in terms of how long they are and their pitch and all kinds of parameters. So I'm not going into details with that, but basically we could try to make predictions what emotions should affect. So for example, if an animal is more aroused, usually what we can predict is that their vocal apparatus will be tensed. So they should have a higher, for example, fundamental frequencies and longer calls, and that's actually what we see. So I'm not going to go into details with that, but I've been writing a review, also looking at what people have found across species, trying to see if there is similar patterns. And then what we see overall, and that has been validated many times. I mean, there is more and more evidence for that. So basically, when an animal is becoming more aroused, which means more bodily activated, so that could be more excited or more stressed. So both positive and negative emotions. So when they're becoming more aroused, as we can predict from the way vocalizations are produced, they will produce sounds that are longer and also more loud and more higher in frequencies. And also they will call at faster rates, so they will call more. So that's quite consistent across species. And then for the valence, it's a bit more tricky to predict. So basically, I've been trying to see if there is common directions in which sounds change when they are produced in a negative state versus a positive state. But usually in positive situations, sounds become a bit shorter. And then that leads me to the last part of my talk, which is the case study. I think I'll have just time to tell you about how we study the expressions and not perception. But basically, what I was interested in this big comparative project between species was to look at similarities between species in how they express emotions. So as I just mentioned, we see clearly that across species, arousal is expressed in the same way, and that's the same between animals and humans as well. So more aroused, which means higher frequency, longer duration, faster rate, as I just mentioned. And basically, we know that it's the same with humans because humans can perceive arousal in the sounds of many different species, from crocodilians to birds to mammals. And then recently, I think it was last year, I also have some colleagues that show that crocodiles also perceive arousal in human babies' cries, which is more for predatory behavior, I guess, but that's show you how conserved this expression of arousal is across evolution. But now with the valence, it was, at least at the beginning of that project that I did, much less clear, because there has been so few studies on comparing calls produced in negative versus positive situations that it was really not clear. It seemed that there is shorter duration in positive situations, but we didn't really know if it was conserved. So that's what we looked at. And to look at that, so to see if there is commonalities in how animals express emotions, you can look at expression. So see how, for example, as we studied all these species, how do they express positive and negative emotions? Or you could look at perception, which I won't have time to show you today, but basically when you play sounds with the speaker, do they perceive the difference between positive and negative calls, and how, and do they perceive this difference in their own species and also across species and from human voice? So the way we studied that, so that's kind of to show you also how we apply the methods I explained at the beginning of my lecture. The way we did that was to try to use similar methods across species. We would always find clearly positive situations. That could be when animals receive food, during positive interactions, and clearly negative situations that threaten fitness, for example, short social isolation or negative interactions like naturally occurring negative interactions. So that's what we would refer to this method 3 I showed you before. So using situations animals avoid or prefer. And then to kind of validate the underlying emotions, in domestic species, we managed to fit a heart rate monitor, especially to know what's the actual arousal of the animal in that situation. And we also assess your behavior to look at locomotion to kind of validate the arousal, assess the arousal, and also validate if we were correct in the valence we attributed to the situations. And then we recorded the sounds they produce, and we tried to see, okay, which parameters of localizations are affected by the arousal that we measured or the valence. So that's all the situations we used. So that could be, for example, anticipation for food, of course, differs between species because they are not vocal in the same situations, but we had social reunion, affiliation, extra for positive, and then for negative, we had food frustration, isolation, all these things. We measured many parameters that I'm not going into details today with, but all the parameters we can to for the vocalizations, such as the duration and pitch and range and frequency modulations. And so for the results, I'm just going to go through, because I guess not all of you are bioacousticians, I will just go through the main patterns. So basically for goats, we found, I don't know if the sound works now, but that in negative states, if it doesn't work, then you can listen to it when I send you the slides. So there are sounds, and you can even see it on the picture, they're much more shaky. So the modulation is much higher than in positive states. So again, it's one same call type, contact call, but it changes depending on their underlying emotions. And then we did a more close comparison of the domestic and wild species, also to see if there was maybe an effect of domestication. So here we compared domestic and Shevasky's horses. Just gonna go show you all of that. But basically, I will play in case you hear them, but negative whinnies, they're very long. As you can, then you can also see it in the images if you don't hear. So they're extremely long, and they have frequencies that are higher and then go down. And then for the positive whinnies, we get very short whinnies with the frequencies that start lower and go up like a different pattern. And then for the Shevasky's horses, we actually see something very different, strangely. So they don't have shorter calls in positive than negative. They have some changes that are the same in terms of parameters between negative and positive situations, but they also have patterns that are opposite. And mainly the main changes we see in the domestic horses, the main indicators of valence, we don't see them in Shevasky's horses. So that's very different pattern, which might be due to the context we use, the individuals, specific individual we use, or the domestication of the species. But if we compare domestic and wild pigs, we actually see something pretty similar, with lots of things in common and lots of things that are very different. So as most species, we see this shortening. So positive grunts are much shorter than the negative ones. And here we see some similarities between the domestic and wild species in terms of these dark lines that you see, which are called the formants. In negative calls, they start very high and then they decrease. While in positive states, they are very flat and much higher in the domestic pigs. And the same kind of pattern occurs in the wild boars, but here we have lower formants in positive than negative states. And formants are very salient parameters, so that's quite a big change between species. And then, so of course, all these results I briefly showed you now, they are basically recorded in very few contexts, as you've seen. It's a lot of work, but it's only isolation versus something else, like 1 or 2 changes. So what we've been able to do only on pigs so far was to see if these changes in pattern can be applied to more contexts and across breeds and also across call types. So not only within grunts, but also if you compare grunts and squeals and screams, can you actually see any pattern occurring? And also, can you train an algorithm, so AI, artificial intelligence, to classify the calls for you, which could be very useful for farmers, right? Because they could just record the pigs and know what's the emotion, the underlying emotion. So for pigs, we've been able to do that through a very large collaborative project called the Project Soundwell. We combined all our recordings and then added some to cover basically the lifetime. So all the contexts pigs encounter in their life, 19 different contexts we had eventually more than 7,000 calls and 400 pigs. And then we've trained a machine learning algorithm to look at the picture. So the picture of the call, and then learn how to differentiate positive and negative sounds. And the algorithm actually managed after training to reach 92%. So it's 92% of the time correct in whether the call was produced in a positive state or negative state. So we kind of read the emotions of the pigs here. And then I will just finish with the kind of the broadening of that kind of machine learning algorithm. So what we've done in most recent years now is to see if we can also train an algorithm to not only differentiate call types of one species, but of many species. And that will basically can only work if we have these similarities between species, as I was trying to reach with that comparative study, so that we actually have some things in common between negative and positive calls across all species. And so if we look at all the species I recorded in these comparative projects I mentioned, so we can see that there is clearly, for most of them, these shorter calls. When they're in a positive state, they usually produce calls with the shorter durations. But there is also the fundamental frequency, so the pitch is also a bit lower and more stable, like I showed with the goats. So in many species, you find that pattern of shorter and lower and less variable frequencies in positive than negative states. And likely thanks to these similarities, a student of mine could actually train an algorithm that works on the extracted parameters of the vocalizations to differentiate between positive and negative vocalizations across basically all these species. So cows and horse, horse, pigs, wild boars, sheep, and goats. So we fed the algorithm with all of these species at the same time. And it managed pretty well, because with 90% accuracy, it can tell us if the call is likely positive or negative. And if we look into more details about the patterns and how they change across species, we do see some patterns like the ones I mentioned with the decreasing duration in positive versus negative contexts, etc. So we do have few parameters that change. So they could be used to as a kind of universal translator of animal emotions. And just to finish off with the advantages of using vocalizations to assess emotions, they're actually very promising because they do indicate clearly arousal or valence. So you can find some parameters that tell you whether the animal is highly activated, and then another parameter that will tell you if this high activation is positive or negative. They're of course non-invasive because we can record animals from a distance. They can tell us the emotion of the animal in real time, not like, for example, hormones that you have to measure in the saliva and take time to actually pass through the saliva. Likely they can be automatized through, for example, machine learning algorithm, as I just showed you. The eye somehow, at least especially for arousal, but also a bit for valence, valid across species. And then, of course, we need more work to know if they are, how precise they are. And of course, they also have limitations, which I have mentioned on these two slides. So they have some issues of, so basically when we look at how good an indicator is, we can look at sensitivity. So whenever we get false negative, so basically when do emotions sometimes change without corresponding change in vocalizations? Of course, that happens quite a lot for many reasons, because vocalizations are influenced by other things than emotion. So they can be influenced by who's around, because they have evolved for communication. So an animal is not going to call when there is no one around. So it might be very stressed, but it might not call because there is no one to hear the vocalization. There might be threshold effects, like the pitch cannot increase more than the vocal apparatus allows. No, sorry, there are threshold effects, which basically will depend on when the animal calls. So it might need a lot of activation to start calling, but also ceiling effects. The pitch cannot go as high as a certain number. And then, of course, there might be differences between call types. And then finally, there are also cases of what we called specificity, so false positive. So here it's when does the vocalization change, but the emotion is not changing. So the internal state is not changing, but the vocalization does. And that, of course, happens a lot because vocalizations can be affected by many factors. For example, the age and sex and body size of the animals. And as I mentioned, also audience effect, which can lead to exaggeration of the call. So an animal might call much more than it is stressed because there is a specific animal next to them. And also it might depend on the function of the call type. And you have to keep in mind that arousal has like a very strong effect, much more than valence. And to finish off, that's just a summary slide of how you can use expression of emotions. So if you want to study that in the future, like for yourself, you can basically ideally put the animals in different clearly negative or positive situations that increase or decrease fitness. Like we've done, you can manipulate the animals if it's a domestic animal, for example. You can, if it's a wild animal, you cannot manipulate, you can just observe when they are having affiliative or agonistic interactions. Ideally, you can measure the heart rate, for example, physiological parameters. You can look at their behavior, and then the heart rate or the movement will tell you how intense is the emotion for the animal. The context could tell you, and the behavior also could tell you about the valence. And then you can record the animal and kind of see, okay, what's the effect of arousal? What's the effect of valence on the sounds that you recorded? And I think I will stop here. I had the last part was on the perception, but I can send the slides and then you can look at what we've done in terms of perception of emotions across species.
Thank you, Elodie. It was really interesting.
Thank you
Does
very
anybody
much.
have questions? I do have some, but I'd like to see if the students. Alguém tem pergunta, pessoal?
It's different. I'd like to thank you a lot for this very exciting.
Yeah, it's wonderful.
I really work on animal behavior, but ecology mainly, and but I think it's so interesting and being deeping very nicely in this field, and it's very interesting how you're doing things. And I think was very interesting to associate these techniques like bioacoustics with the emotions of the animals. And I'm quite interested in the differences you found between wild and domestic animals. Very, very interesting this, because the domestic ones you can't control, but the wild ones, no. This is a big difference, but I mean, you found very differences in terms of acoustic sounds.
Yeah, we found many differences between the. I mean, of course, again, it's like limited. We had slightly different contexts. I mean, there are many things that differ more than the domestication itself, but both with the domestic and Przewalski's horses and the pigs and wild boar, we find clear like opposite patterns. The most interesting one being, I think, this change in formants between the wild boars and the pigs. So basically, pigs, when they are producing a happy grunts, the formants are higher, while wild boars, when they produce the equivalent of happy grunts, the formants are lower. And that's a big change. And when we played wild boar, so the part that I didn't have time to show, but I will send in my slides, where basically we played pig calls to wild boars and wild boar calls to pigs. And then we check if they perceive difference between the positive and negative calls. And interestingly, the wild boars, they reacted with behavior suggesting that they were in a negative state when they were hearing positive pig calls. So when they hear positive pig calls, they freeze and they do all these things which indicates negative emotions. And that might mean that could mean many things, but it could mean that they perceive wrongly the emotions of the pigs. So they misinterpret the emotions of the pigs.
That's very interesting. We are doing, so my project, we are trying to see the effect of tourism on the river dolphins, and I'll very much likely enter in contact with you when we're doing more analysis, because what we wanted to do is see if somehow they communicate stress through their sounds in relation to the interaction with tourists. So we are imagining like how, like if tourists touch them or just offer them fish, or slap the water, if they caress the dolphins, like, oh, we're taking all these behavioral methods. The only caveat that we have is that we can individualize the sounds of the dolphins. But I was looking mostly at how noisy the calls were, measuring like entropy and harmonicity, and seeing if we have differences in, as well as calling rate. But now with this form, and I never thought of duration, because mostly all their calls are super short. So I don't know if duration is a good measure for them, but that's very interesting. And the formant part as well. I'll try to look at that. And if you don't mind, I would like to get in contact with you later and see what are your thoughts and if we can collaborate as well. And what we did last year, we captured them. We took them out of water to do exams and collect samples. So when they were isolated on land, we also recorded the sounds that they were producing. So we would have more like, they were definitely like negative emotion sounds. That you could see that the animal was like, not comfortable. So that would be nice to look into those sounds as well and see, if we can define what is a more negative. Of course, it's more extreme, right? But it's definitely a negative emotion. What these would look like for a dolphin.
Yeah, that's great, because there's so little work on dolphins.
Yeah.
On bottlenose dolphins, I'm aware of one PhD thesis that I assessed where they looked a bit at indicator of arousal, and I think was valence as well. But if you have these two contexts, that's perfect, because you have two negative states, and one that is a bit less aroused, I guess, and one that is much more when they're outside of the water. And so I would measure definitely, duration and rate, and and frequencies, and entropy usually increases in more aroused states, but it really depends on the species. It's not always the case.
Yeah.
And formants, I don't think you can see formants, right, in dolphins, because they're very high pitch, right? But maybe
yeah, river dolphins are not so high pitched. They're really, they are lower in frequency than the marine ones, so we might be able to see some,
nice.
depending on the calls. Yeah, and and when we were writing the proposal, I remember I struggled so much because there were barely any studies, and basically the things that people looked for stressors like calling rate and like the noisy parts. And I only, as you said, I only found like a few cases more talking about when dolphins were being translocated, or and it's or they were sick. And I have a friend of mine in the US that she did some stuff with bottlenose as well, Brittany. I don't know if it's the PhD you assess, but
no, that was someone from Australia. I can find.
Yeah, and I reviewed one of her papers. I never seen the published thing in the end, but she also looked at, but basically calling rate was one of the indicators of welfare and stress in dolphins as well. But it was very interesting. I really enjoyed your talk.
Thanks.
And have all these things to look for. I'm excited. Yeah.
I think there was a question in the chat.
Oh yeah,
There is.
Oh, he's asking if if you could identify these patterns of emotions, negative and positive emotions in different species, because he has a bad microphone.
Yeah. So so there are a few things, as I mentioned, that we can find. So we can find mostly similarities in arousal. So that's super clear, and it's also logical because it's linked to vocal production. So when animals are more tense, you can easily predict what happens to the vocal apparatus and hence to the emotion, to the vocalizations, I mean, like longer calls and all these things. And for valence, it's much more difficult because it seemed to be much more species-specific kind of codes. But eventually, we still see some patterns that are similar across species. So that's this shortening of vocalizations. So usually shorter vocalization in positive and negative states, although apparently not in humans. But also lower frequencies and more stable, less shaky calls in positive states.
Yeah, it's very interesting. Yeah, that's true. Oh, you got like a shaky voice when you're nervous.
Yeah, also, yeah. It's. I mean, for the valence, it doesn't seem to map much with what we find in humans. Mostly, I have colleagues working on non-speech sounds like laughter or like screams. You could have screams of pleasure or screams of fear, and then if you compare these, you actually find something quite different than we see in animals. But it's interesting also.
It's super nice.
I have one question, a very, very strange question, about the killing when the man is going to killing the pigs and cows. And I think this study, your study, is very important because the animals should be very relaxed and before will be killed, like because the production of cortisol or something like that is very high if they are very stressed, I don't know if they use the methods to do the animals to be relaxed and excited or something like that. You know something about this?
So I mean, there's lots of potential applications of these tools. Like that's what we were aiming at with the peak project Soundwell, but we never reached that stage. But nowadays there is, especially with the AI tools, there is more and more possibilities to maybe make an actual tool that farmers can use, and maybe we can use in slaughterhouse. To make sure animals are not too stressed. But especially on farm, because there is lots of monitoring tools for health, to know if a cow is limping, how much cows are eating. But there is nothing, from what I'm aware of, there is nothing that monitors their mental health or their emotions. Although nowadays people clearly consider emotions as the most important part of the welfare of animals. Because an animal can be sick and, have medication, but if it's depressed, it's much worse than if it's sick and happy, if you want. I mean, that's at least what welfare researchers consider nowadays. But there is no system on farm that can monitor emotions yet. So using vocalizations would be one, but it's quite tricky because what we've done is to extract calls by hand and then use AI to classify them. But do the extraction is extremely difficult, and so far we are failing with with that part. There is no even most advanced AI tools yet cannot find the exact beginning and end of especially low frequency grunts of the peaks, for example. Maybe in the future, yeah.
Okay, thank you.
What do you use to extract your calls? Like, what kind of software or anything? I've used in the past, like, Artwarp in MATLAB, and I've also used Lucinia, like, to extract, like, the contours or parameters that I want.
Yeah. So in my group, we use many things. I mean, I mostly use Prat, and then I usually prefer to do things by hand because sometimes things are very noisy and an automated tool cannot find the exact beginning and end of the calls. But I had, so for example, my postdoc Evelyn, she's been developing our package called Chunk and what was the name of her package? Chunk and Feed, I think, and it's like finds based on the amplitude, the beginning and end, and then it processes and it sort out. I mean, it puts in different folders the calls as you want. So I have students that have been using R also for the analysis, for example, SoundGen, that is very good tool for extracting parameters. And then we've been using also Audacity for labeling sounds.
Yeah, I'm doing everything by hand as well because it's hard to. When you water what is already noisy and then you have a noisy call, it's hard to do things automatically.
Yeah.
Very interesting. Mais alguma pergunta, pessoal? Nope.
Não tem aqui no.
Yeah, we don't have any more in the chat.
Yeah.
But then, if we don't have any more questions, I would like to thank you very much, Elodie. I really enjoyed your talk. It was very, very interesting, and
Thank you.
I hope we can collaborate more in this line of work. It's so nice. Thanks a lot.
And
Thank you very much.
thanks. I will send you the PowerPoint. It might be very heavy, so I might have to send you a link to download it, but I'll find a way, and then you can share it.
Thank you.
Thanks.
Thanks a lot.
Bye.
Bye. Have a nice evening.
Thank you very much.
Okay.
Bye-bye.